Eosinophils were once known only for their role in allergy and parasitic infection. Today, we're uncovering their far-reaching influence in immunity, inflammation, and beyond.
Eosinophils are specialized white cell. While they participate in immune responses, they are also key players in allergy and asthma and in other chronic inflammatory diseases. New data suggests eosinophils may also play pro- and inhibitory roles in many other areas, including tumor development and neural growth.
Understanding eosinophil biology is essential, not just for treating these conditions, but for advancing our overall understanding of the immune system itself in both health and disease. That's where the International Eosinophil Society steps in.
IES is a unique platform where clinical research meets basic science. Our members are driving discoveries that move from the lab bench to the patient's bedside.
What sets our society apart is our seamless integration of disciplines. By bridging clinical practice and foundational research, we unlock new possibilities. IES is more than just a meeting. We're a society of individuals of all parts of the globe, interested in using science to best understand the eosinophils and rapidly apply that to the benefit of patients with eosinophil diseases.
IES has provided me with invaluable opportunities to connect, learn, and grow. Through the society, I've met mentors, collaborators, peers who have profoundly shaped my career. It opened doors I didn't even imagine, allowed me to share my research globally and find mentors beyond my university who truly believed in me.
IES values collaboration, mentorship, and different perspectives. We bring together people from around the world with different backgrounds and scientific expertise. What sets IES apart is the generosity of the research community. We share reagents, experimental models, ideas, and support to advance science through trust and teamwork.
I didn't realize how valuable this network was until I attended my first IES meeting. It gave me access to science I hadn't seen before and the chance to show my own work with experts I never thought I'd meet.
As an early career member, IES has helped me grow both professionally and personally. It's a community where I felt supported, challenged, and inspired by the work and the people around me.
At IES, we're not just advancing science. We're building a future where eosinophilic diseases are better understood, better diagnosed, and better treated. If you're passionate about eosinophils in the lab, in the clinic, or anywhere in between, the IES is your community. Join us and build that future.
Hello and welcome to our IES webinar today. My name is Jessica Gates. I'm joined by my co-moderator, Matt Drake. We've got some excellent speakers for you who we're really excited to hear from. But just a few housekeeping bits to start with. So just go through a few slides. So firstly, I'm sure most of you are very well aware about the International Eosinophil Society, and you just watched the promotional video, which was beautifully made. But you will all be aware that there are biannual meetings, monthly webinars and other educational programming available through the International Eosinophil Society for everyone who's interested in the eosinophil. And on that note, if you're not yet a member but would like to be, we'd encourage you to join the IES, and you can do so through this QR code. At the moment, there's a limited time offer for 50% off regular membership. So if you know of any colleagues who aren't yet members and who would like to be, then please point them in this direction. And also just a reminder for trainee members, this is free for all pre-doctoral and postdoctoral individuals. So if you've got anyone in your lab or in your clinic who might like to join, then please encourage them to. And one of the big benefits of being a member of the International Eosinophil Society is the conference, which will be next year in Vancouver. I attended my first congress last year, and I certainly am hoping to be there again next year. It was a really wonderful event, lots of opportunities for talking to people and collaborating and sharing ideas. So I would encourage you to all put this in your diary. And then before we've even started today's webinar, this is just a plug for the next webinar with some excellent speakers looking at type 2 inflammation across diseases, eosinophils, IL-5 cytokine and unmet needs. So put that in your diary for Wednesday, the 6th of May. And with that, also a note to our sponsors. Thank you for their support. Without them, we wouldn't be able to run these events. So thank you to all of them. And then finally, just some housekeeping and how this is going to work today. So I would encourage you all to submit questions through the question and answer button anytime during the presentation, and then we will address them at the end with the speaker, which we will read to them. You will all be muted and you won't be able to chat, but you can post your questions in the Q&A. This is all recorded, and the recording will be put on the IES website afterwards. And video and closed captioning and live transcription are available through the CC button. So with that, I'll hand over to my co-host.
Thanks, Dr. Gates. And good morning, good afternoon, good evening for some of you. Thanks for joining us today. It's my distinct pleasure to be able to introduce a fantastic speaker lineup today. We have a theme today centered around innate immune mechanisms that control eosinophilic inflammation. And our three speakers are going to cover all elements of these concepts. Our first speaker is Dr. Kelly McNagny, who's going to be discussing neonatal microbiome alterations and lifelong susceptibility to eosinophilic asthma. Our second speaker is Dr. Lisa Spencer, who's going to discuss gut-derived innate lymphoid cells and eosinophils disseminate type 2 immunity along the gut-lung axis in food allergy. And finally, our third speaker is Dr. Caspar Ohnmacht, who will be discussing metabolic control over the innate lymphoid cell Tuft cell circuit and eosinophilia in the gastrointestinal tract. Without further ado, let me introduce in more depth our first speaker, Dr. Kelly McNagny. Dr. McNagny earned a bachelor's in biology and biotechnology from the Worcester Polytechnic Institute and a PhD in cellular immunology from the University of Alabama at Birmingham, where he studied B-cell maturation with Dr. Max Cooper. He completed postdoc work at EMBL in Germany with Dr. Thomas Kraft, focusing on stem cell fate and blood cell development. And he later established his lab at the University of British Columbia, where he is now a full professor in biomedical engineering and medical genetics. His research centers on stem cell biology, immunity, inflammation, cancer, and immunotherapeutics, using transgenic models and high throughput technologies. He's held national leadership roles in major research networks and currently co-leads the immunotherapeutics cluster at UBC and serves as the UBC's delegate at the Canadian Institute for Health Research. Welcome, Dr. McNagny. Thank you for being here.
Thank you very much for that terrific introduction. I hope I can do that one justice. Listen, folks, I really wanted to thank the IES profusely for the opportunity to talk. This is a terrific webinar series, and it's become a must-see for my lab. So hoping most of them are tuned in today to make sure I get the stories right. Today, what I really wanted to do is give you our view of how immune responses are initiated, but more so how immune responses are sculpted during development and regulate whether you're going to be susceptible to future inflammatory disease. And I'll use this as an example, eosinophilic asthma, but then towards the end, I'll just point out that I think this is a more general concept for a whole variety of immune-mediated diseases. And so let's see if I can get us a pointer. And here we go. So overview for what I wanted to tell you about today is just give you a very brief introduction to our view of immune response and show you kind of a family portrait of lymphocytes that cover a spectrum of responses. Tell you about some of the subspecialties of those lymphocytes and the role of innate lymphoid cells in particular and regulating organ-specific immunity. And then I'll dive a little bit more deeply into how innate lymphoid cells regulate eosinophilic disease and use the example of allergic asthma as an example of a concept we've developed called immune sculpting during development. And I'll point out that we have a parallel pathway for Th17 immunity, which involves ILC3s and IL-17 and neutrophilic disease. Won't have time to talk about that today, but I'll give you a plug to some of that work towards the end. So very briefly, for my entire career, beginning, when I was in Max Cooper's lab, most immunologists have been focused, laser focused on these two cell types, B2B cells and alpha beta T cells. And that's for good reason. These are really the targets of every vaccine program in the world. And that's because if you get immunity from these cells right, you'll be protected lifelong from disease. B2 B cells are exquisitely antigen specific. They'll protect you against one and only one infectious organism if you get the immune response right. Same for the T cells. Now, despite that focus, if you and I were all dependent exclusively on these cells to keep us from getting sick, I think we'd probably all be dead. And the reason for that is, despite the fact that these cells are really exquisitely specific and make a really robust immune response, they're slow to develop. It takes a good week to 10 days to develop a good B cell and T cell response to a new organism. And when you've got an infectious agent that's doubling within hours, there's no way they can keep up. And what people tend to, and a good example actually of where we could all be dead is if you look at COVID, that's a great example of a new infectious agent that came along. And if you look at people who died from COVID disease, really at the end stage of their disease, they had perfectly fine B cell and T cell responses. The problem is they just came about too slowly or were hyperactive. Now, what people tend to forget is that, in fact, there's a whole series of similar lymphocytes that develop much early in development. And I'll use an example of B1 B cells. These are cells that come up during fetal development. They make antibodies just like B2 B cells, but those antibodies tend to be lower affinity and very broad specificity. And you could ask, why is that important? And I think the answer is that when you come out of that sterile environment of your mother and into this sewer of antigens that we're exposed to, having cells that have a broad specificity and maybe a lower affinity is really important for protection, slowing things down just long enough so you can make a more specific response later. So that's for B1 B cells. But I'll also point out that we have a similar cadre of T cells, mate cells, mucosal-associated innate T cells, and KT cells, and gamma delta T cells. Again, these receptors on these cells tend to be broader specificity, recognize classes of pathogens rather than very specific pathogens, and just slow down that the infectious organisms long enough to develop a more protective immunity later. Coming back to more recent cells, so within the past 10 to 15 years, we've identified a whole new class of lymphocytes called innate lymphoid cells. These cells are even more broad specificity in that they don't even have antigen receptors. Instead, these cells use alarmin type receptors. They recognize patterns of damage to tissues and then design the appropriate immune response to deal with that damage. And that's kind of what I want to focus on today. Now, getting more granular, clearly you folks are aware that we have Th1 T cells for fighting viruses, which make things like TNF-alpha and gamma interferon, Th2 T cells, which make the intermediate number cytokines, if you will, IL-4, 5, 6, 10, and 13. These are designed to help us fight parasites, detoxify toxins, and make us feel like hell when you've got an allergic response. And then you have Th17 cells, which are more potently attuned to fighting bacteria and fungi. And I just want to point out that not only for those innate type T cells, the early T cells I showed you earlier, but even for the innate lymphocytes, we have a parallel cell type for every one of those T cell sublineages in the innate compartment, regulated by these series of cytokines. Now, are those cells important for local tissue immunity? Oh, and one other thing I wanted to point out. These cells come up very early in development. They colonize tissues, and then they become largely tissue resident thereafter, expanding and contracting in response to damage. And probably the reason it took us so long to find these cells is, number one, they're fairly rare, and you only see them if you look in organs. They rarely circulate. And I think that because we tend to look more in blood to find immune cells, that's one reason why I think these cells were missed for so long. Now, are they important for immunity? Let's take lung as an example. And if you look at an allergen exposure, virtually all allergens contain proteases. Those proteases do damage to epithelia. The epithelia releases alarmin-type molecules like IL-33, TSLP, and IL-25. Those alarmins activate innate lymphoid cells, which are subepithelial. Those cells instantly respond by making things like IL-5 to recruit eosinophils, IL-13 to activate dendritic cells and cause them to mature. Those mature dendritic cells then grab any antigen they find in this milieu, traffic to the lymph nodes, and begin the process of stimulating antigen-specific immunity by polarizing Th2 T cells. Those cells then produce factors that cause isotype switching of B cells to things like IgE and IgG1 in mouse. Those cells then recirculate through thoracic duct lymph and colonize the organ that's become inflamed. The IgE coats basophils, which then become long-term resident cells with lots of antibody on them. That's why you can generate an immediate response the next time you're exposed to an antigen in the same milieu. So that's our model of how immunity occurs in the lung. Are the ILCs important for that? Well, here's an example we showed many years ago where they are. Here we've knocked out ILC2 selectively by deleting a transcription factor called ROR alpha, which is critical for their development. And we've run these mice through a house dust mite model of inflammation. And what you can see is in wild-type mice, you get lots of infiltrating inflammatory cells in the parenchyma and around the airways. And in the ROR alpha knockouts, the ILC2 knockouts, it's hugely attenuated. For the eosinophilic folks, here's looking at eosinophils staying with major basic protein, and you can see massive numbers of eosinophils in the lung in the house dust mite model, and a real huge attenuation of eosinophil infiltration in the mice where we lack ILC2s. Now, this is just looking at total inflammatory cells in the bowel fluid. You can see there's a huge attenuation of total inflammatory cells, but again, most of the attenuation is in the eosinophil compartment. You fail to get good recruitment in the absence of ILC2s. But importantly, it's not just the innate response, it's actually adaptive response too. So without the ILC2s, you fail to make antigen-specific antibodies very effectively, and again, you attenuate inflammation. Same is true for the T cell compartment. You have much less IL-4, IL-5, IL-13, actually also IL-17, which is likely important for evolution of the immune response later in asthma. Okay, so what I've shown you so far is if you get rid of ILC2s, you really turn off the tap right at the very beginning. You tend to block innate responses as well as the downstream adaptive responses. Now, I want to shift to something a little bit different, and this is in the development of allergic responses. How is an immune response sculpted during early development? Now, it's been known for a really long time that there are a variety of environmental factors that make you more susceptible to allergic disease. Some of the things that are protective are older siblings, dogs, natural childbirth, breastfeeding, farm living, exposure to helminth infections. All of these things tend to be protective and prevent you from developing allergies early in life, at least epidemiologically they do. And then there's a variety of risk factors. They make you more susceptible, and they're the counteract factors. C-section, formula feeding, high-dose antibiotic use, multiple courses before the age of 2 makes you much more likely to become allergic. And I think it's pretty obvious that all of these things influence your exposure to microbiome. And many years ago, we decided to set up a model where we could look at that antibiotic exposure, see if we could get a handle on how that makes you more susceptible to allergic disease. And the model we developed was very simple. We put pregnant dams on low-dose antibiotics, and the goal here was to make sure that the first exposure of the pups to a microbiome was an altered microbiome that came from their moms. We then put those mice, continued them on antibiotics, and then waited until they were adults and looked at their susceptibility to allergic disease in this ovalbumin model of allergy. Now, if you look at lungs without induction of allergy, they look nice, lacy lungs here. If you expose them to ovalbumin, you get lots of inflammation around the airways and around the vessels. Streptomycin, low dose, again, not enough to change the number of bacteria, but to change the subtypes of bacteria. Didn't have any effect on allergic asthma model. But vancomycin had a huge effect. It made the mice much more inflamed and increased the susceptibility to allergies. And this is also true for germ-free mice. And this is some beautiful work from Kathy McCoy many years ago, showing that germ-free mice will get allergic to just about anything you expose them to, whether it be through the skin or the airways or the gut. Now, interestingly, that's not true of just general inflammation. So here we've run mice through a model of hypersensitivity pneumonitis, a Th17-driven disease. It's a T-cell alveolitis. This is a model of farmer's lung for those of you who want to be more specific. When we run mice through that model, we find that here we're showing inflammation in response to farmer's lung antigen. With vancomycin, we see no difference in the immune response with those mice, but now instead with streptomycin, we get a much more severe disease. So the take-home message here is that different shifts in biota early in life make you more susceptible to either a Th17 or a Th2 disease, and it's antibiotic dependent and microbiome dependent. So how does it work? Well, first we qualified the window of opportunity to have that kind of effect. So we wanted to know when the susceptibility is established. We tried putting mice on antibiotics through pregnancy and up to weaning and then taking them off, allowing them to get recolonized with a different flora, and then check 7 weeks later as adults how they respond to ova. Or we didn't treat them with antibiotics and only began antibiotic treatment after weaning, and then 7 weeks later induced asthma. And if you do that type of experiment and you take a look at when the susceptibility is established, here's what we see. So this is looking at total inflammatory cells in the lung. With ovalbumin, you get a certain threshold level of inflammation. With vancomycin, that's more severe, as I showed you earlier. With streptomycin, it's not any more severe. There's no difference. That's with neonatal antibiotics. But if you do the same experiment where you're exposed to antibiotics late in life, it has absolutely no effect on susceptibility. So it looks like there's an early life window of opportunity to sculpt an immune response that later in life makes you more susceptible to disease. Now, looking at how that's established. Here, this is a complicated slide, but basically each individual bar here is an individual mouse, and we're looking color-coded at the repertoire of bacteria the mice have in their gut. And what you see is with control mice, we have a broad spectrum repertoire. With streptomycin, that's shifted somewhat. And with vancomycin, it's dramatically shifted. And the major shift here is that we're losing the bacteria that metabolize fiber into short-chain fatty acids like butyrate, propionate, and acetate. So those bacteria get lost with vancomycin treatment. Looking at our mice, what we find is if we look in the feces, we find selectively butyrate is down in the mice that were exposed to vancomycin, whereas acetate and propionate didn't change. Now, butyrate is really interesting because it's an HDAC inhibitor, so it's perfectly poised for altering epigenetic gene expression in progenitor cells. And in fact, some of you folks, old-time folks, remember treating cell lines with butyrate in order to induce them to differentiate. It's a really good epigenetic modifier. So our question was, okay, if we're taking away bacteria that produce butyrate, and that's all that's driving disease, then what we ought to be able to do is, in addition to adding antibiotics, put back the butyrate and reverse the susceptibility. And that is exactly what we see. So here, if we take mice that were on regular drinking water and we induce ova-induced asthma, we get a certain threshold of inflammation. If we now add butyrate back, that doesn't change. If we look at mice that are on vancomycin, we get, again, exacerbated disease. And if instead we put them on vancomycin plus butyrate, we get a complete reversal, and now they're right back in register with the wild type, the normal mice. So it looks like we can reverse with just that one metabolite. So, quick summary here. Antibiotic-driven shifts in flora make you more or less sensitive to Th2 inflammatory disease. We show that this correlates with a drop in bacteria known to produce short-chain fatty acids, we can give them back in trance and suppress. And altering the microbiome early in life can have a long-term and lasting effect on immunity in adults. And that's true for kids too. It's well known that if you look at the microbiome of kids in a big cohort study we did in Canada several years ago, you can find selective alterations and a decreased complexity in the microbiome if you look at 3 months in kids. And that very well predicts which kids are likely to develop allergies by age 5. Okay, now, showed you this slide before. This shows you our pathway for inflammation in the lung. The question is, are any of these cells altered by treatment with butyrate or by loss of microbiome? So we had a look at that more recently. So let's start with the ILC2s, which are right up at the apex. So we looked in adult lungs of mice that have been on antibiotics. Here's how we recognize ILC2s. They're lineage negative. They express the IL-7 receptor. If you look in the lungs of mice that were on antibiotics, you find that you have twice the number of ILC2s in the lung of the adult mice compared to ones that were on regular drinking water. And that's true of ones that are ST2 positive or GATA3 positive for the aficionados. But the bottom line is you have more ILC2s. If you look at the production of IL-5 or IL-13 from those cells, you find a much higher level at steady state of IL-5 production from the ILC2s in mice that were exposed to vancomycin, and the same for IL-13. We also find that those ILCs now make autocrine IL-2, which is causing them to proliferate more. So to summarize that small bit, with antibiotic exposure, cells now make more IL-5 and more IL-2, IL-13, and they make autocrine IL-2, and there's a higher number of those cells at steady state in the lung. How does that change immune response? Well, if we take those same mice and we prime them with IL-33 for just a couple of exposures right before and analyze the immediate responses to IL-33 as an alarmin, here's what we see. We find that you get an enormous increase in the amount and the number of ILC2s. So now you get a really big boost in the number of ILC2s in the lung immediately after exposure to IL-33. And we also find that in contrast to normal mice, which normally don't make ILC2s in the lung, normally don't make immediately IL-4, it takes many exposures to induce the IL-4 production. Now these mice are instantly making IL-4 the first time they get exposed to an alarmin. That's quite different from regular mice. So what that shows you is that now in response to alarmins, we get more IL-5, more IL-4, and a further expansion of ILC2s. What do these cytokines do downstream? So I mentioned to you we have two subsets of B cells, B1 cells, which make more innate type broad specificity antibody, B2 B cells, which make more exquisitely specific antibody. We looked at both of those compartments in the lung. And what we found was that with IL-33 in the mice that were exposed to vancomycin, we get a rapid switch to IgE production from these mice. So these are mice that are making antibodies, IgE antibodies from B1 B cells, which are broad specificity. And this just shows the amount of IgE that we get from the B1 cells. It's a huge amount compared to the control mice, which are on regular drinking water. So IL-5, in addition to expanding eosinophils, is also expanding the B1 B cells. These are one of the few B cell subtypes that makes an IL-5 receptor. So they're expanding. They also are responding to the IL-4 by class switching to IgE. And now we're getting natural IgE produced from those B cells. And for the aficionados, this antibody that we're finding from these B cells binds things like phosphorylcholine, a whole variety of things that are very broad spectrum. What's downstream of that natural IgE production? Well, we also had a look at the basophils in the lung. So here I'm showing you IgE bound to the surface of basophils in regular mice exposed to regular drinking water or mice that were exposed to vancomycin. And what you see is Fc epsilon receptor binding of IgE on the surface at this threshold level. But with vancomycin, it's about 5 to 10-fold higher on those basophils. So these basophils are absolutely coated with natural antibody that will bind to a broad slate of antigens. And this just shows you the intensity here. In the interest of time, I'm just going to go over that quickly. But we get now more basophils coated with a high level of IgE, and they're just sitting waiting to respond to any antigen that crosses the epithelial barrier. We also see an expansion in terms of mast cells in the lung, in terms of the total number of mast cells, but also their frequency. Same is true for eosinophils in either the lung parenchyma or in the bowel fluid. So we're starting a cascade that expands everything for an allergic response. And so the bottom line is, this is the cascade we think we've established by treating mice with antibiotics early in life. More ILC2s, they're primed for reactivity, they expand the eosinophils and recruit them more rapidly, they cause isotype switching to make natural antibody, and that leads to all the downstream effects you would need for priming an allergic response. Because the antibody is a natural type of antibody, we think it causes the mast cells and the basophils to degranulate to virtually any antigen they get exposed to. And this is why the mice and probably kids are more susceptible to allergies if they have an altered microbiome. Now, you could use any inhibitor you want along this pathway to try to dampen this effect and improve disease in kids or in mice. But I'd mentioned to you that short-chain fatty acids seem to be a great way of blocking this early in life. So we did exactly that. We went back and added short-chain fatty acids to our vanc mice and then had a look at this cascade. The bottom line is, if you look at that enhanced IL-5 production, IL-13 production, and IL-4 production, simply adding back the butyrate completely reverses that and brings these mice right back in register. Now, this is a Th2 story for allergic asthma, eosinophilic asthma, but I wanted to point out we have an identical story for ILC3s in the lung and hypersensitivity pneumonitis. It's ILC3 driven. It's a different bacterial metabolite that drives this disease, and taking away this inhibitor with exposure to streptomycin makes mice more susceptible to hypersensitivity pneumonitis, also neutrophilic asthma, and that's a story that I can tell you about another time. So overarching summary for this talk, there are a range of unexplored lymphocytes other than traditional T cells and B cells that protect us from disease or are defective in disease. Some of these are long-lived and sculpt a long-term immune response that makes you more susceptible to disease. Distinct neonatal microbiomes have remarkably selective effects on your future susceptibility to disease. And this really changes the way you think about disease. It makes you more focused on neonatal life as a window of opportunity for perhaps preventing you from being susceptible later. I've shown you that for allergic asthma. I've mentioned we have the same story for hypersensitivity pneumonitis, which is ILC3 dependent. Same thing for Crohn's disease. It seems to be ILC3 dependent. And I'm just predicting that you're going to find similar things for things like multiple sclerosis, type 1 diabetes, and rheumatoid arthritis in the future. And those are things we're looking at now. If you want to learn a bit more, here are some papers we published very recently. This is a Crohn's disease model in JXMed. Here's the hypersensitivity pneumonitis model in mucosal immunology. Everything I have shown you has been focused on organ-specific inflammatory response, but I'll also point out that ILC2s reside in the bone marrow, as do ILC3s, and we're now exploring their ability to skew hematopoiesis centrally. And I think they also play a role in driving disease. And I'll just say, stay tuned for the story from Tae Kim on balancing eosinophilic versus neutrophilic asthma with ILCs. And Maggie Chopra in my lab is doing a beautiful story on the clonal development of ILCs, which we think are T cell or thymus-derived. They're an offshoot of early thymic progenitors, and how those relationships are developing during development. And quick plug for the ILC6, I already mentioned that. And this is my lab. Most of this work was done by this crazy guy, Ahmed Kabil, terrific scientist. He's now working with Marco Colonna at Harvard at the Broad Institute. Mike Hughes, my long-term postdoctoral fellow, just took a job in industry, and I'm still mourning that. And this great international and really diverse group that's my lab and have put their faith in me. These are the guys that make me look good. They're the ones who do all the work and really deserve all the credit. And with that, I'm happy to take questions.
Fabulous. Thank you so much. What a tremendous story you've brought together here. We have time for probably one question here, and then some answers in the Q&A can occur through the rest of the talk. So Dr. Denberg asks first, what effects do antibiotics such as Vanco have on eosinophil, basophil, and mast cell differentiation from marrow progenitors?
It's a terrific question, Judah. And I'll tell you, so we just put out a story, just came out, where we've been looking into that. And what we find is that those exposures I showed you, we can take the bone marrow precursors from those mice, transplant them in recipients, and we transplant the susceptibility to disease. Now, there's two possible ways that could work. That could be epigenetically modifying the stem cells in ways that make them more likely to spin off eosinophil progenitors and basophil progenitors and a lot of things. Klaus Nerlov at Oxford has a really nice story that I think parallels ours and suggests that is part of the story. But the other part is, don't forget, ILCs colonize the bone marrow and they live there long term. You have them sitting there and they're geared towards making IL-5 and 13. I wouldn't be surprised if we've epigenetically modified those cells as well, and they make you more likely to spit out eosinophil progenitors. So it could be both of those effects in the bone marrow. But it's a terrific question, and we're looking into that.
Fantastic. In the interest of time, we're going to move on to our next speaker, but thank you very much. And there's a couple other great questions in the Q&A if we want to get to those in writing throughout the rest of the talk.
Thanks, everybody.
Thank you. Our next speaker is Dr. Lisa Spencer, who will be talking on gut-derived innate lymphoid cells and eosinophils disseminate type 2 immunity along the gut-lung axis in food allergy. Dr. Spencer is a faculty member at the University of Colorado School of Medicine and serves as the scientific director of the Gastrointestinal Eosinophilic Diseases Program at Children's Hospital in Colorado. Her research focuses on the immunobiology of mucosal tissue eosinophils in the airways and gastrointestinal tract, with the goal of developing improved therapies for eosinophil-associated diseases. Her work has led to key discoveries in the field, including mechanisms of eosinophil degranulation and evidence that extracellular eosinophil granules remain functionally active in tissues. Dr. Spencer is well known to the IES and one of the leaders of this organization. We're very excited to have her talk today. So welcome, Lisa.
Thank you so much. Find my slides here. So thanks again for hosting this session and letting us talk about these items. Is my camera on? Can I ask, Matt?
It is not.
Interesting.
There it is.
Sorry about that. All right, so thank you so much. I'm going to pick up a little bit where Dr. McNagny left off, in that we're looking at the allergic march. After all of those other factors that in prenatal development and the environmental factors and all of the things that act on us to be more susceptible to these allergic diseases, we want to ask the question, once all of those influences are there and you've got some sort of an allergic disease at any of these sites, whether it's a food allergy response or atopic dermatitis, what is it about that inflammation, that allergic inflammation in that specific site along the skin, gut, lung axis that makes you more susceptible to develop another allergic disease along a different site? So we're really trying to understand what the crosstalk between those different mucosal organs are in individuals who are already allergic. And so because of that, we're using a model, we're going back to this very old ova-alum model where we use adjuvanted ovalbumin over 2 weeks to really sensitize these mice to this egg protein. And we think that this mimics a strong Th2 biased immune response that should model our patients kind of coming into this arena. And so then upon that background, where we can make a very standard sensitization across the board, we want to ask the question, what happens if we then challenge those patients, or those mice in this case, at any of these different sites along the skin, lung, gut axis? So we'll follow that sensitization with either an epicutaneous sensitization on tape strip skin, which is a model of atopic dermatitis, or we will do an oral gavage and put this antigen directly into the stomach for a food allergen model. And what we also did in this paper that I won't talk about this morning is we did intubate the mice and also drip it into the lung, so we could have a lung exposure model as well. And then we waited 4 days and just asked the question, what happens at all of these different sites, the skin, the lung, and the gut? And what we found is, as not surprisingly, you get a strong eosinophilic inflammation at whatever site you put the antigen on. But you also change your eosinophil resident population at both the lung and the gut. And so what we mean by that is I'll show you 2 examples. Here is from the skin challenge. So these mice received 3 skin challenges on day 21, 22, and 23. And then we looked at their lungs on day 27. And I think you can appreciate from where the arrows are here, we see more eosinophils in the lung. And this is not an eosinophilic inflammation. We rarely even see 2 cells touching each other, but it is either a doubling or a tripling of these cells, depending on the experiment. And we also noted that those eosinophils are actually much closer to the airways than they normally are. So in a healthy wild-type mouse, you very rarely will see a lung eosinophil anywhere near the airway, whereas after these skin challenges, or intragastric allergen challenges, we really start to see eosinophils near the airways. They also are CD62 low, and they seem to have an inflammatory phenotype. And the same is true here, if you can see the quantification on the far right of those after intragastric allergen challenge, so a food allergen challenge, we see the same increase in lung eosinophils. And if you look at those lungs a little bit more carefully, you can start to see that it looks like there's mucus in some of those airways. So again, there's no allergen here. We did not put any of this allergen into the lung, and we don't see an expansion. We don't see an IL-4 response. We don't see alarmins. We don't see any evidence of allergen being in the lung, but we do see this increase in mucus-positive airways. And in collaboration with Chris Evans, he helped us out looking at the different mucins that are now produced there. So at the top, you have your healthy mucins, which are MUC5B, but you can see after the food allergen challenge here, your lungs start to shift to a MUC5AC, or an inflammatory mucin, and you can see evidence of fucosylation from that pink stain there. And so this really made us wonder, okay, if the lungs look like this, are they actually more susceptible now to inhaling something else, a totally different allergen? And so this case, we followed this sensitization and challenge with an intranasal house dust mite regimen. And this is similar to what Dr. McNagny just told us, except instead of taking this out the 3 weeks that in our hands we really need to expose mice to HDM in order to actually get a response, we only did it for 2 weeks. And instead of using the 6 micrograms of HDM we normally use, we dropped that down to 3 micrograms. So this is a suboptimal dose that does not induce inflammation. And what we found is if you have a normal mouse that did not have a food allergen challenge and you give them this suboptimal HDM dose, their lungs look absolutely fine. But if you first challenge that mouse, a sensitized mouse with ovalbumin, to their gastric tissue, then they have this massive cellular infiltration in the lung tissue. And if you look at the bowel fluid, they have a significant increase in bowel leukocytes. So we're inducing an allergic airways-like response to an otherwise innocuous allergen. So we got very excited about this being one of those mechanisms as to why perhaps some patients are more susceptible to a second allergic disease after having a first allergic inflammation at another site. And so this day 27 is where I've been showing you the data of eosinophil inflammation, mucin dysregulation, and overall lung priming. And that was work that was done by Courtney Olbrich in our lab. Some new studies I'll show you now are all from Natalie Falta, who is an exceptionally talented student in the lab. And this work is in a paper that she has under revision right now. And so we asked the question, if all of these things are happening on day 27, what do these 4 days look like in the lung? And we can look at IL-5 and IL-13 because these are obviously common eosinophil cytokines. And so IL-5 does go up even with sensitization, and that's been shown by other groups as well. It's both in the lung and the adipose tissue. But IL-13, surprisingly to us, we had a very strong signature, and this was happening certainly with 1 day, and I'll show you some data that it happens more quickly than that. And it doesn't happen with challenge alone, and it doesn't happen with sensitization alone. We also looked at all of the alarmins, and I won't go into this. Dr. McNagny just taught us about these. None of the alarmins are going up. None of the alarmin receptors are going up except IL-17RB. And this is really what caught our attention. And the reason for that is our parasitology colleagues. And this paper came out a number of years ago from Richard Grenz's group, where they could show if they gave a parasitic helminth infection, in this case, they're using Trichinella spiralis, gave this to the intestinal tract, they saw a mucus secretion in the lung. And again, this is an intestinal dwelling nematode. It did not crawl through the lung. And they could show that this was because an inflammatory ILC2, so it's another subset of ILC2s, that's IL-25 responsive, got turned on in the gut, trafficked up to the lung, spit out some IL-13, and that's what's driving the mucus secretion. And so when we think about now a new subset of ILC2s, of this inflammatory ILC2s, how do they differ from the natural ILC2s? Well, Dr. McNagny just told us that these natural ILC2s like to sit in the tissue and stay there over time. Inflammatory ILC2s like to leave. And so rather than being ST2 positive or responsive to IL-33, they tend to be responsive to IL-25. So they express the IL-25 receptor, IL-17RB. And they, again, they're migratory and very, very strong IL-13 producers, and they don't produce very much arginase 1. And so our question then became, is this why we actually have more eosinophils coming into these remote lungs after a food allergy response? And so we teamed up with Lee Reinhardt at National Jewish because he was an ILC2 expert, and I learned very quickly you need some help when you're trying to stain for ILC2s. And so we came up with a staining panel, and we're basically gating on these lineage negative cells that are gated here that are KLRG1 high. And I will say, if you're looking for a natural ILC2 that's ST2 positive, they're going to be at about this level of KLRG1 expression. And so we do actually see what we feel are inflammatory ILC2s in the lungs of these mice. And this is actually happening within 1 hour of getting that intragastric allergen challenge. And you can see it here shown as the total frequency, as well as the total number of inflammatory ILC2s. And for these total numbers, we take the entire left lobe and digest that. We do also see natural ILC2s, they're ST2 positive and KLRG1 intermediate, but they don't tend to go up significantly there. And so the question then becomes, is this like the intestinal model? Are these also coming from the gut, or are they somehow coming from either the bone marrow or the lung? And we think it's the gut. And the reason for that is, first, when we look by flow cytometry, we can detect them in the duodenum. And it's important that it's the duodenum. We actually don't detect them at this 1 hour time point in any other region of the small intestine. It's just the duodenum. We can also see them within the draining mesenteric lymph nodes. And we know that they traffic to the mesenteric lymph nodes following an S1P gradient, and that was published by several other groups. They also leave the mesenteric lymph nodes, get into blood circulation, and go to the lung from an S1P gradient. So we asked the question, if we blocked that gradient and trapped them in the intestine, would we also lose our inflammatory ILC2s in the lung? And in fact, we do. We lose them in both the mesenteric lymph nodes and in the lung. So we think this is fairly strong evidence that the inflammatory ILC2s that we see in the lung after a food allergen challenge really are coming from the small intestine, and in this model, specifically the duodenum. And so then becomes the question of, well, how are they being generated? So in the parasite studies, it's really a Tuft cell mediated response. And so Tuft cells are these chemosensory cells that sit in the epithelium, and they monitor mainly bacterial metabolites. And it's been shown now that parasites can kind of come in and take over this process. There's also some data now out from Richard Loxley's group showing that the regular nutrient pathways can be detected here. But there really hadn't been, to our knowledge, direct evidence of food allergens interacting with the Tuft cells. And so they could be interacting directly, or they could just be modulating this microbiome. And we've just learned how much the allergic response really does modulate the microbiome. And so we asked, first of all, if Tuft cells are important. And in fact, 1 hour after this intragastric challenge, again, only in the duodenum. We don't see this in the jejunum or any other region of the small intestine. We see a nice induction of a Tuft cell gene signature. It's difficult to detect much of a significant increase in the total cell numbers, so we really do need to understand how significant this is. But the signature is certainly there, and it certainly correlates with inflammatory ILC2 signatures. We also see a nice induction of IL-25, again in the duodenum, but not in the jejunum. And so our question then became, well, we're seeming to be inducing Tuft cells. We're definitely inducing IL-25. Is it because these food allergens are just dysregulating this microbiome? And so the metabolites that are acting on the Tuft cells are what's causing this expansion. So to do this, we repeated our model. So here's our systemic sensitization. But on day 21, before we gave the food allergen challenges, we collected fecal pellets and performed shotgun metagenomics. And we do find some differences. If you look at the phyla level, there's not really much here to write home about, but you do see a difference in the shift of the firmicutes to Bacteroidetes ratios there. And so that is in line with what an allergic response should be inducing. But if you go all the way down to the species level, you really start to see beta diversity as well as alpha diversity in these between these two conditions. And I think it's probably easiest to see in that panel on the far right, where you can really see a reduced complexity in the bacterial species that are in those guts. So we really did expect that this would impact the Tuft cells, looking at some different succinate producers, and that could be what was driving our response, but we were wrong. So if we look at our Tuft cells, whether you count them by flow cytometry and look at the total cells, or if you look at their gene signatures, the change, this dysbiosis in our model was not enough to induce that Tuft cell expansion. We also didn't see any IL-25 at the protein or the RNA level, and we don't see any signature or cells or expansion of these inflammatory ILC2s. So we do have dysbiosis, but it is not what is expanding the Tuft cells, inducing IL-25 secretion, or expanding the inflammatory ILC2s in our model. And so we really asked the question then, food allergens may be interacting directly with Tuft cells, and we still need to answer that, and we don't have that answer yet. But a more important question to us was, could we just avoid it altogether? Can we take allergen out of the equation? Can we look at IL-25 as a potential nodal cytokine that could drive this whole response and still see all of the downstream effects? And in fact, the answer is yes. And so we treated the mice for 3 consecutive days with IL-25 and then looked at their lung tissues and intestinal tissues. By day 3, so 1 day after that final IL-25, we see a nice induction of mesenteric lymph node inflammatory ILC2s. We also see the lung inflammatory ILC2s coming 1 day later at day 4. And this correlates, if you look at the IL-17RB as a surrogate for inflammatory ILC2s and compare that to IL-13, it really is a strong one-to-one correlation in that secretion. And if you look at the same parameters, here's the lung inflammatory ILC2s or the lung IL-13, in the presence of FTY, so again, this is the S1P modulator that's going to block their migration out of the gut and into the circulation, we see a loss both of the lung inflammatory ILC2s and the lung IL-13. So we think IL-25 really does recapitulate what we were seeing with allergen, and it avoids all of the other off-target effects. And it also gives us the same kinetics. And so really you see a nice induction of these inflammatory ILC2s peaking in the lung on day 4 of this IL-25 protocol. And this is really 1 day before you begin to see eosinophils at their peak level. So this is similar to the day 27 of the food allergy model. And if you look at those eosinophils, once again, at the top here, this is the vehicle treated. In the bottom, you have your IL-25 treated. You get the same distribution. So they're close to these airways, within 50 microns of the airways in ways that you just don't see in these untreated mice. And that's quantified over here on the right. And again, the majority of these newly infiltrated eosinophils appear to be CD62 ligand negative, which is one of the markers of an inflammatory eosinophil from Claire Mesnel's work. I also want to note that this drop-off that you see in the inflammatory ILC2s, others have shown that this is because they convert to natural ILC2s, and we see that in our model as well. So here's the natural ILC2s measured after IL-25 treatment at that day 5 time point. So you've got eosinophils and natural ILC2s in the lung at this point. But if you use mice, and again, this is a collaboration with Lee Reinhardt's group, if you use mice that are deficient in BATF, which is a transcription factor that is necessary for inflammatory ILC2s, but not necessary for natural ILC2s, you really see that you lose those natural ILC2s. So these natural ILC2s that we do see, along with the eosinophils, likely are coming from these migrating inflammatory ILC2s. And so what are the inflammatory ILC2s required for? It seemed like a slam dunk. We know ILC2s recruit eosinophils, so we assumed that they were required for the mucins and they were required for eosinophils. We were right on part of that story. We do see here, if you treat in a food allergy model with OVA and look at the day 27 lungs, I've shown you that MUC5AC transition. We can show that if we take the inflammatory ILC2s away, that's this last bar here, we lose that MUC5AC. However, we don't lose the eosinophils. So in this particular case, we still have eosinophils and we block the inflammatory ILC2s, but we just argued perhaps the food allergy model is activating other things. Maybe natural ILC2s are coming from other places. So we took the allergy out of the equation and repeated our allergen-free IL-25 model. Once again, if you block the inflammatory ILC2s, you do not block the eosinophils that come into the remote lung. So here we argued maybe it's an off-target effect of FTY720. We know that eosinophils express S1P receptors, and perhaps we're trapping the eosinophils. So we used a third model and went back to this genetic model. So again, these mice in purple are basically wild-type mice. The ones in blue are BATF deficient, so they don't have inflammatory ILC2s. We can show once again that you get MUC5AC transitions here after IL-25 treatment, and you lose that if you take away your inflammatory ILC2s. However, again, we did not block the eosinophil migration when we took away the inflammatory ILC2s, so they're not necessary for eosinophil remote homing in any of these models, whether we deplete them pharmacologically or genetically. So, it's a summary. We treat with a food allergen, and that's going to elicit Tuft cells, IL-25, and inflammatory ILC2s. And I've labeled this the small intestine, but for this model in food allergy, we're really seeing this as a duodenal localized response. And we like this idea of IL-25 being the nodal cytokine here. Again, we can bypass all of this, the dysbiosis and the Tuft cells, as long as we provide the IL-25. These gut-derived inflammatory ILC2s are migrating to the mesenteric lymph nodes and then to the remote lung and driving lung IL-13 expression within 1 hour of that intragastric challenge. And when you take these cells away, you really lose that IL-13 response. Within 1 day, they're converting to natural ILC2 phenotype, so they're still secreting their IL-5 and their IL-13, but they no longer look like an inflammatory ILC2. And these are the cells that are going to coexist with this at the time where you have this mucus expression and MUC5AC dysregulation. They also are going to coexist with the eosinophils that come into the remote lung, but again, they're not connected. Maybe they're sufficient to induce the eosinophil infiltration, but they're certainly not necessary for that. So by 3 to 4 days after the food allergen challenge, you can really see this peak of eosinophils. They're localizing near the airways. They're contributing to this heightened airway responsiveness. And so with that, I will thank again the folks that did the work. So Courtney Ulbrich took the lead on the mucosal immunology paper that first showed the remote priming effect. And Natalie Falta is the lead on this current work that I've just shown. And Leah Larson has done an incredible amount of that work as well. And Chris Evans, again, is our collaborator with the mucins in lung information, and Lee Reinhardt and an exceptionally talented postdoc in his lab, Isik Khan, have really been instrumental in helping with the inflammatory ILC2 in recognition of the process. So thank you so much.
Fabulous. Thank you, Dr. Spencer. Maybe I'll lead off with the questions quickly here. We have just a few minutes, so please drop your questions into the Q&A. I'm fascinated by the fact that the gut and the lung have this axis that you've really uncovered in remarkable detail. But is it that the homing of the or the ILC2s and subsequent eosinophil expansion, is that specific for the lung, or is this going to be a systemic effect, and we're just happen to be looking in the lungs and seeing expansion there? And if it is specific to the lung, why? What's driving it?
Yeah, I would say I don't think it is specific to the lung. So how many other organs, which other organs, and under what circumstances still need to be worked out. But from preliminary data that we're seeing now, I don't think it's going to be specific to the lung. It's a great question.
Couple questions in the chat. Did you happen to look to see if the inflammatory ILC2s also migrate to the bone marrow and alter eosinophil poiesis?
That is a great question, and it's on our list. So no, we have not done that yet, but we are very excited about it. And especially, thinking of some of what Dr. McNagny just talked about as well, I do think it's important to start looking at what is affecting hematopoiesis, what could be inducing some epigenetic changes in those eosinophils.
There was also a question about the model. And during a food allergy challenge, does the physiological response to allergen result in any inhalation of allergen to the lungs, thereby contributing to driving the lung eosinophilia?
That's a great question. So we did a lot of dye studies in the beginning to really make sure that we weren't aspirating. I don't think it's possible to really know that you never aspirate. We also, we always check the cytokine profiles, and if we get a super high IL-4 spike, we toss the mouse out of the experiment because we assume we've expanded Th2 cells. But I want to stress that we did all of those studies with the epicutaneous model as well. And that's really what convinced me, because I couldn't come up with any way that the mouse was going to have it inhaled from the epicutaneous model. And what's interesting from Raif Jiha's group a number of years ago from Boston had shown that just the tape stripping, just that mechanical disruption of the keratinocytes can drive an IL-33 response in the keratinocytes that acts on the gut to secrete IL-25. So I think we're still finishing those experiments, but I think both the epicutaneous and the intragastric are going to be hitting that nodal IL-25 and driving the whole response.
Wonderful. In the interest of time, we'll move on to our next speaker, but thank you, Dr. Spencer. Wonderful talk. So our next speaker is Dr. Caspar Ohnmacht, who will be speaking on the metabolic control over the innate lymphoid cell Tuft cell circuit and eosinophilia in the gastrointestinal tract. Dr. Ohnmacht earned his PhD in immunology from the Ludwig Maximilian University in Munich, Germany, working on basophils and eosinophils in the context of allergy and helminth infection. He then did a postdoc at the Institut Pasteur in Paris, France, working on tolerance mechanisms towards the intestinal microbiota. And then since 2014, he's been a principal investigator of the mucosal immunology group at the Center of Allergy and Environment at the Helmholtz Munich and Technical University in Munich, Germany. Welcome, Dr. Ohnmacht. We're excited to hear your talk.
Sorry now. Can you hear me now? Yes?
We can hear you. We can't see you.
You can see me. Okay, please. So just a second. So now it should work, right?
Now we can see you, but we've got your slides. There we go. Perfect.
Okay, perfect.
You see your slides, we can hear you. Thanks.
Okay. So thank you very much. I mean, it has been a wonderful webinar so far, and I'm really excited also to share one of our stories. And most important things first. So the story I would like to share with you today is the one of Carsten Huth, so a very talented PhD student who recently graduated. And he really made some interesting observations. So I'm very happy that Dr. Spencer already introduced the Tuft cells and the ILC2s already so well, so I don't have to spend so much time on this. And in contrast to both of the previous speakers, I will talk about the beneficial effect of type 2 immunity and also ILC2s in the context of a parasitic infection. And the model, as you will see, that we use is this bug here. It's a parasite called Nipostrongylus brasiliensis that is able to induce a really strong type 2 immune response. How we came to this story, for that reason, I want to introduce to you shortly the aryl hydrocarbon receptor, or AHR for short. I'm sure you may have heard of that. It's quite an important transcription factor that is localized in the cytosol, and only upon binding of a specific ligand, it translocates to the nucleus and regulates gene transcription in lymphocytes, but also many other cell types. We got interested into this transcription factor because we made observations that the HR plays quite a major role in the context of allergic diseases, such as in macrophages, but also in lung epithelial cells, for instance. So what catched our interest is that there are different sources of these small organic compounds that serve as ligands for this aryl hydrocarbon receptor that can be derived from pollutants, but also can be generated in the skin, for instance, upon UV radiation, and also can be derived from the diet or from gut microbes that produce some of these substances. However, also the host actually is able to generate its own ligands for this transcription factor, and they are mostly derived of tryptophan. So they are tryptophan metabolites, such as indoles, kynurenins, and kynureninic acid. And the enzymes that catalyze these reactions are IDO1 and IDO2, so they have been very well characterized, but there is a recently emerging enzyme that is called alpha-1. So what are these enzymes? So both IDO1 and alpha-1 are inducible L-amino oxidases that are primarily expressed by myeloid cell types. And as I already mentioned, IDO1 was already well studied and is quite good understood, whereas alpha-1, even though it has been discovered already more than almost 30 years ago now, the function of this gene is still not very known. And the name is derived from the discovery in B cells after IL-4 stimulation, where Paul has first cloned this gene. And what these enzymes do is that they catalyze the reaction from tryptophan into kynurenine or N-formylkynurenine in the case of IDO1, whereas alpha-1 can convert phenylalanin, tyrosine, and also tryptophan, so all aromatic amino acids, into their respective keto acids, namely phenylpyruvate, 5-hydroxyphenylpyruvate, and indole-3-pyruvate. And one major difference between both of these enzymes is that IDO1 is stored within the cell, so within the myeloid cell or the B cell, whereas alpha-1 is a secreted enzyme that has therefore the capability to shape the local microenvironment. So now I want to shortly introduce the model because maybe not all of you are familiar with this model. So N. brasiliensis, that's a natural mouse and rat parasite that drives a very strong innate and adaptive type 2 immune response that is naturally cleared within 10 days in a wild-type mouse. And the way this infection works is actually that larvae penetrate the skin. So we mimic this by injecting the parasite into the skin. Then within 6 to 18 hours, by the bloodstream, the larvae reach the lung, where they develop further into another larval stage within 18 to 72 hours. And then they are penetrating actually into the alveolar space. They are caught up and swallowed. So meaning that starting from day 3 to 4 onwards, you can find them in the small intestinal tract where they mature. Usually they mate, they lay eggs, and then the eggs are released and then the cycle starts again. And if you can see here in the small intestine, you can see here, these are the adult worms. They are quite long. They are macroscopically visible. And here in the lung, you can see this larva here, an L4 stage larva that is localized in the lung. And very interestingly, during this infection, both in the lung and also in the small intestine, we see a very strong induction of these enzymes, so alpha-1 and also IDO1. I will only speak today about alpha-1 because we found no effect whatsoever with IDO1 and IDO2. So therefore I will concentrate on this alpha-1. And the question that we first addressed is actually whether the parasite can itself induce the expression in dendritic cells, like a myeloid cell type. And for that, we generated bone marrow-derived diseases, treated them with an extract of these parasites, and then measured alpha-1 expression. And indeed, this extract was almost as efficient as another TLR that is able to induce alpha-1 expression in dendritic cells. And that was true not only on the transcription level, but also on the protein level, because you can see here, by using a self-made polyclonal anti-mouse antibody, that this extract induced very rapidly the expression or the protein expression of alpha-1 in a tissue lysate. And then after 24 hours, it is also released into the supernatant of these cells, indicating that the parasite can directly induce alpha-1 expression, at least by dendritic cells, possibly also in other myeloid cell types. And you can also visualize this if you block the release of these vesicles, and then the alpha-1 will accumulate actually in the vesicles of bone marrow-derived dendritic cells. But what about in vivo? We then infected both wild type and alpha-1 deficient animals and looked and established a staining to detect alpha-1 intracellularly by flow cytometry. That took quite some time, but in the end, Carsten managed to do that. And as you can see here in wild type animals, you can see that there is alpha-1 expression, whereas in the knockouts, there is no staining whatsoever. And as you can see here at steady state, we already see in the mesenteric lymph nodes, there is a basal expression of alpha-1 in these conventional dendritic cells. And this is increased upon infection with N. brasiliensis. And also the same is true for small intestine. Here at steady state, we barely detect any alpha-1 expression, but there is a massive induction upon infection. And we were also able to identify alpha-1 staining, presumably in dendritic cells, in histology sections of Nipostrongylus brasiliensis infected animals. I hope you can see here that these alpha-1 positive cells are scattered along the epithelial cells here, but we also detect occasionally alpha-1 expressing cells within lymphoid follicles in the duodenum of these infected animals. And this is also validated actually in our own single cell sequencing data, where we can detect CCR7 and alpha-1 co-expression in conventional dendritic cells of the mesenteric lymph node, but also of the small intestinal lymph node. But this is really true for all publicly available single cell data sets that we looked at. So there is always a very high correlation between activated dendritic cells, in our case indicated here by CCR7 expression, and alpha-1 expression. And this is an example here from Meyer et al., where they could really show that there is they call it enriched in regulatory genes dendritic cell subset. In our view, this is always an activated dendritic cell subset, where always alpha-1 is part of the signature of these activated dendritic cells. And interestingly enough, this is always associated slightly with a Th2 response in this case here. Okay, what we then looked is whether these alpha-1 deficient animals do have any defect in clearing N. brasiliensis. And this was really a striking finding, because most animals really can clear very well this parasite, and you really need a severe deficiency really to fail that the parasites are cleared in animals. But here you can see, even though similar numbers of parasites reach the small intestine, the alpha-1 deficient animals actually fail to control the parasites and then also fail to clear the parasites by day 10 of infection, indicating really that alpha-1 plays a major role in control of these parasites. And along the infection, actually, you can look, because there's a very strong type 2 immune response really induced systemically, so including the lung. And you can see here that also lung eosinophilia is severely impaired in alpha-1 deficient animals. And that really goes along also with a deficiency in the generation of typical type 2 cytokines in the bronchoalveolar fluid, such as IL-13, IL-4, and IL-5, indicating that we also have an impaired type 2 immune response in the lung. We then also tested the small intestine. And here again, the effect was even more striking. You can see there is a massive eosinophilia that is induced in the small intestine of wild-type animals, whereas alpha-1 deficient animals barely induce any eosinophilia after infection. And that goes along again with an impaired induction of typical type 2 cytokines like IL-4, IL-5, IL-13 here on the transcript level of total tissue, but also on the protein level for IL-13 here, indicating that also in the small intestine there is a severely impaired type 2 immune response. And as it has already been introduced, I want to quickly remind everyone that there is really a cycle, a positive feedback loop between ILC2s and Tuft cells. And as already indicated, Tuft cells are really a unique source of IL-25 in the intestinal tract, because Tuft cells are the only cell type that expresses IL-25. And IL-25, along with other alarmins such as IL-33 and also some lipid mediators, is able to activate and expand intestinal ILC2s. And this ILC2-derived IL-13 is then able to act on intestinal epithelial stem cells or some progenitors to induce the further differentiation of Tuft cells, indicating that there will be a positive feedback loop to really drive the whole cascade of innate type 2 immune responses. As already mentioned before, currently I think there is no direct evidence that Tuft cells directly sense helminth infections, whereas they can sense some they have chemosensory receptors that can, for instance, sense succinate that is released by some protozoans. And, however, I'm sure that this audience is very well aware that ILC2s are absolutely essential for eosinophilia. So because, for instance, here the study by Christoph Klose et al., they really showed very nicely that ILC2s are absolutely essential as a source for IL-5 and also for eosinophils, both at steady state, but particularly after inducing eosinophilia with various challenges. So therefore, we also looked, of course, for ILC2s, which is not so easy in this animal model because there's a lot of mucus production and it's quite tough to get alive cells out of the lamina propria of the small intestine. But Carsten managed, and we can really see that ILC2s barely expand in IL-4/1 deficient animals, whereas we have a nice expansion of ILC2s after infection with Niposongulus brasiliensis. And also, we measured these alarmins, so we can see that IL-25 is induced massively, whereas IL-4/1 knockout animals fail to induce IL-25 increase after infection. And the same is actually true also for IL-33 and also TSLP, indicating really that this cycle is severely impaired in the absence of IL-4/1. And to really make sure that the source of this IL-4/1 is really derived from the hematopoietic compartment, we generated also bone marrow chimeras where we transplanted IL-4/1 deficient bone marrow into wild type animals and vice versa, waited around 10 weeks to make the immune system restore completely, and then infected these animals. And again, we really found that it's really hematopoietic IL-4/1 that is absolutely essential to enable control of the worm count of these parasites and also to induce expansion of ILC2s, indicating that it's really hematopoietic IL-4/1 expression is absolutely required to mount a robust innate type 2 immune response here. And as we already talked about Tuft cells, we then also looked for Tuft cell markers. So by gene expression profiling, we identified that indeed DCLK1 expression severely goes up after infection, whereas this was not the case for IL-4/1 deficient animals. And we were also able to really visualize this and quantify this. And as you can see here, around 10% of wild type of the epithelial cells in wild-type animals seem to be Tuft cells here, indicated by DCLK1 staining, whereas there is barely any increase in IL-4/1 deficient animals. And finally, we will also manage to stain for Tuft cells directly ex vivo. And you can also see here, also by this quantification, that the Tuft cell expansion is severely impaired in the absence of IL-4/1. And to now address the question whether we can bypass the need for Tuft cells, we injected IL-25 into animals. And this really indicated to us that the expansion of ILC2s was completely normal. So there was no problem of ILC2s to expand in the absence of IL-4/1 when there was a sufficient source of IL-25. And that also allowed now to control the parasites again, to expand ILC2s after infection, and also to expand the Tuft cells now, indicating that when we bypass the activation of this cycle between ILC2s and Tuft cells, then we can completely restore the immune response. And finally, we also measured the specific metabolites that are induced by IL-4/1, and tryptophan, as I mentioned, is converted by extracellular alpha-1 into indole-3 pyruvate. And indole-3 pyruvate, by the way, is a very potent ligand for the aryl hydrocarbon receptor. Indole-3 pyruvate is typically incorporated into cells and unfortunately not very stable, but rapidly further converted into indole-3 lactate, indole-3 aldehyde, or indole-3 acetic acid. And when we measured some of these compounds by metabolomics, you can see that the levels of tryptophan were completely normal. You can see that kynurenine is completely dependent on the expression of IDO1 and IDO2 as expected, because this pathway was already well known. However, the only compound where we found a difference was indole-3 lactate, because wild-type animals can induce an increase in indole-3 lactate upon infections, whereas alpha-1 deficient animals completely fail to do so. And for that reason, we wondered whether the aryl hydrocarbon receptor also plays a role in this response. And our first idea was that maybe the Tuft cells directly express the aryl hydrocarbon receptor, and we tested this with an IHR tomato reporter mouse. And indeed, as you can see here, Tuft cells do express the AHR, at least on a transcript level. So there is also non-epithelial cells that do express the AHR receptor. And we currently, to be honest, cannot really say whether the alpha-1 derived metabolites do act directly on the epithelial cells or not. But with that knowledge here, we at least can say at least the Tuft cells do express the receptor, and there is the possibility. And when we look in complete AHR deficient animals, we can see that also here, these animals have a non-controlled parasite count. They also fail to expand ILC2s. And for the Tuft cells, the picture is not so clear. There seems to be a slight reduction, but we're not 100% sure. The problem is that many cells in the gastrointestinal tract do, in fact, express the HR receptor, which was nicely published by Dini et al. Already in this publication here, which I can highly recommend if you're interested in intestinal HR expression. And to sum all of this up, our current working model is that somehow these parasites are sensed not by Tuft cells, but by myeloid cell types, such as dendritic cells and possibly also macrophages. They get activated. They secrete alpha-1. This alpha-1 converts tryptophan and maybe also other aromatic amino acids into specific metabolites, including indole-3 pyruvate and indole-3 lactate as a consequence. And that may regulate HR activity in different immune and also epithelial cell types, possibly. And then this will in the end, induce the differentiation of Tuft cells, the release of IL-25, expansion of ILC2s, secretion of IL-13 to further drive this cycle, induce eosinophilia as well. And IL-13, of course, from ILC2s is also necessary for mucus production and goblet cell hyperplasia, which are both really highly important features to drive expulsion of the parasite from the host. And with that, I would like to stop. I would really like to thank my team that was quite instrumental for all of this work, and also our collaborators. Thank you very much.
Wonderful. Fantastic talk. We're open for questions for the next few minutes. Feel free to drop questions in the chat or Q&A. Maybe I'll lead things off here. It seems like there's a few steps between understanding aryl hydrocarbon receptor activation and subsequently Tuft cell activation. What do you speculate are the connections there between, if it's not a direct effect on activation of Tuft cells, if this is a secondary cell type that's being activated to subsequently induce IL-25 release? What do you think are some of the potential mechanisms that are driving that effect?
Yeah. That is an excellent question, and we are actively working on that. We have tried to, establish intestinal organoids to assess whether the differentiation of Tuft cells directly depends on HR expression, for instance, or can be manipulated by external alpha-1 supplementation. But to be honest, we don't have a clue yet. And the problem is really that almost every cell type in the gastrointestinal tract expresses AHR, including ILC2s and also ILC3s. And one candidate that we have in mind is actually ILC3s, because there was a publication where it was shown that rank ligand is also necessary to boost Tuft cell differentiation. And this rank ligand may be derived from ILC3s, which we know depend on HR expression for the release of other cytokines. So that may be one hypothesis that we follow, but we don't have an answer yet, and we still need to work on that.
There are obviously in the lungs, there are similar kind of Tuft cell, functional Tuft cells and whatnot. Do we think that this is actually a process that may be mirrored in the lungs, given the life cycle of these parasites?
Yeah, that's an excellent question, and we don't know yet. So that's on our list to do, that we also look for the role of IL-4/IL-1 in the lung. I personally think, for sure it will be expressed in the lung, because I think that's pretty consistent that all activated dendritic cells seem to express really IL-4/IL-1. What's the consequence of that in the lung, I think, is not so clear yet. I think this we need to study, because the cycle between Tuft cells and ILC2s, I think, is most established in the gastrointestinal tract, and it may be a bit more complicated in the lung, and there may be additional factors that impact on that.
Excellent. Well, if I don't see any further questions, I'll say thank you again to all our speakers for wonderful talks today. I'll turn it over to my co-moderator, Dr. Gates, for some closing remarks.
Thank you. And yes, just to echo that, a huge thank you to all three of our speakers today, Professor McNagny, Dr. Spencer, and Dr. Ohnmacht. Really fascinating talks, and really nicely linking the gut and the lung, as well as ILC2s and eosinophils. So we've crossed lots of different parts of what the IES is all about. So with that, I'd like to thank all of you for attending as well, and also to the IES team for facilitating today's session. Please, can you scan the QR code and give us some feedback, and there'll be an email as well, I believe, for feedback. So we would be grateful for that. And if you have any other suggestions about topics you'd like to hear about, do let us know. Hopefully we'll see you all again in May for the next webinar. And yes, please have a lovely rest of your day wherever you are. Thank you.